Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were discussing about cellular signaling, in which we have covered multiple pathways that are critical for cellular functioning. Today, we are going to discuss about NF-kB signaling, which is a slightly different signaling pathway and a very unique pathway compared to others. So, in last class, we learned about hedgehog signaling. We have also learned about Wnt signaling. Both of them are quite similar, but the NF-kB signaling is very different from those two signalings. Like in the case of Wnt and Hedgehog signaling, when the cell is at rest, means the signal is not arrived, such as Wnt protein or Hedgehog protein, they are not arrived at the site. In this condition, the transcription regulator of those pathway, that is ubiquitinated and subjected to the proteasomal degradation, so that it is cleaved, and the cleaved party and the cleaved protein, the cleaved part can work as the repressor for the transcription of the genes, for the regulation of transcription of the genes. So the responsive genes will be shut down when the signal is not there. But in the case of NF-kB signaling, the signaling differs in these terms, that the transcription regulator, it is present there, but it is inhibited by the inhibitor, and this inhibitor will be ubiquitinated and will be degraded, so that the NF-kB is released inside the cell and it can do its job. So this is the difference between the Wnt signaling, Hedgehog signaling, and the NF-kB signaling. So today we are going to learn about NF-kB signaling. So NF-kB, it is the acronym for nuclear factor kappa light chain enhancer of activated B cells. Okay, so this NF-kB, this gets activated in response to either bacterial infection. So when cell is exposed to bacterial infection, NF-kB will get activated, or it can get activated when the cell is subjected to viral infection, or it can even get activated when there's inflammation due to the cytokines, the inflammatory cytokines such as TNF alpha or interleukin 1, IL-1. Okay, so those inflammatory cytokines, they can bind to their receptors, so that they will activate NF-kB downstream. So these are the factors which exist upstream of this NF-kB signaling, NF-kB factor, so that they can activate NF-kB. The other stress that can activate NF-kB is ionizing radiation. So stress conditions such as ionizing radiation can activate NF-kB, so that the cell gets ready to handle those stress conditions. So in all of those conditions, the different types of receptors, they are going to get activated. For example, when there is bacterial infection or viral infection, the factors over the bacterial cell wall, they are going to bind to the toll-like receptors. So the toll-like receptors could get activated in the mammalian cells, or the toll receptors in the Drosophila. Okay, so those toll-like receptors get activated and they are going to activate the NF-kB. So these are the four different situations that are going to activate NF-kB downstream. So once this NF-kB is, it is how it is activated. Let's see. So there we are going to discuss two scenarios where the signal is absent, means when those stress conditions are absent in the cell, means cell is at rest, and when those stress conditions are there, what happens to NF-kB, that we are going to discuss. So when the cell is at rest, first condition is that cell is at rest. Okay, so when cell is at rest in the cytoplasm, let's assume that this is a cell. So inside the cell, there exists nucleus, right? So here we can see the NF-kappa B. Okay, so NF-kappa B, so let me just change the color. So this is NF-kappa B. Okay, so NF-kappa B is heterodimer. So it has two monomers and they are bound. They share homology at their N-terminal domain, and that is why they are bound together at their N-terminal domain. So those two monomers that are going to form the NF-kappa B are P65 and P50. So both of them, they share homology at N-terminal domain. So at their N-terminal, they are going to bind to each other and they are going to form NF-kappa B. So this NF-kappa B, so this particular part is N-terminal domain where the P65 and P50 they are bound. So let's assume that this is P65 and this is P50. So this is the NF-kappa B molecule, and when cell is at rest, it is bound to its inhibitor known as I inhibitor of kappa B alpha. So this protein is the inhibitor which is going to keep the NF-kappa B in its inactivated state when the cell is at rest. So this also binds to this NF-kappa B at the N-terminal domain. So here it binds. So this is the inhibitor of kappa B alpha. So this protein binds to this NF-kappa B at N-terminal, and at this N-terminal there is nuclear localization signal. So that this nuclear localization signal sites, they can be, they can be hidden, they can be bound, or they can be occupied by this inhibitor, so that NF-kappa B stays in the cytoplasm in its inactive form. Because this NF-kappa B, this is a transcription regulator, means this is also a latent transcription regulator, means this is going to go inside the nucleus to do its downstream job, to activate the genes, the NF-kappa B responsive genes, which code for cytokines or chemokines, so that the cell is able to escape, or it is able to handle the stress condition, or when the infection is there inside the in the tissue. Okay, so that is why this is how the inhibitor of kappa B, it is going to keep the NF kappa B inside the cytoplasm in its inactivated condition. Okay, so this is what happens when cell is at rest. So let's see what will happen when the cell, okay, when the cell is exposed to the stress conditions that were mentioned here. So when the cell is exposed to the stress condition, maybe it could be um, it could be the bacterial infection, it could be viral infection, or it could be ionizing radiation. In all of those cases, a three protein complex is going to get activated. Okay, so whenever the four different condition that I mentioned before, they are seen in the biological system, the three protein complex in which the inhibitor kappa B kinase exists, this three protein complex is going to get activated. So this is inhibitor. So this is inhibitor kappa B kinase. Is I kappa B kinase. So this means that it is going to phosphorylate the I kappa B inhibitor, so that the NF kappa B is released inside the cell. So what happens is that, let's assume that this is the cell surface, and over the cell surface different receptors, let's say that this is the toll-like receptor, this is toll-like receptor, which is going to receive the bacterial or the viral cell wall components, or there could exist the receptors for TNF alpha. So this is TNF alpha receptor, this is IL-1 receptor, Okay, so in all those conditions, those different receptors, they are going to downstream activate a three protein complex. Okay, so this has inhibitor of kappa B kinase, its beta subunit, inhibitor of kappa B kinase alpha subunit. Okay, so this is a kinase complex, okay, and it is bound to a protein known as NEMO. Okay, so this is the NEMO. This is NEMO protein. NEMO. Okay, so this three protein complex will get activated in all of those conditions. So the in all of those conditions, the subunit that is going to get activated is the inhibitor kappa B kinase beta subunit. So IKK beta subunit, it is going to get activated. So it is also known as the converging point for all of the extracellular signal. So no matter whether it's ionizing condition, whether it's bacterial infection or viral infection, or the state of inflammation where the TNF alpha is being secreted by some other cells or IL-1 is being secreted, in all of those conditions, the subunit of the kinase complex which is going to get activated is the IKK beta. So this beta subunit is also known as the converging point for all of the extracellular signals that are present, that could be present, which are going to downstream activate NF-kB. So what happens is that this beta subunit, this will get activated. So this gets phosphorylated. Okay. So this gets phosphorylated, means this is now activated. So now this kappa B subunit is going to phosphorylate the I kappa B alpha, okay, which is bound to the NF kappa B. So this was our NF kappa B. Okay, and to this NF kappa B, the inhibitor kappa B is bound, right? So this phosphorylated beta subunit, this activated beta subunit, it is going to phosphorylate this kappa B. So the phosphorylation of kappa B will result into the, so now this kappa B is phosphorylated. The phosphorylation will be done at two serine residues. Okay, so the beta subunit is going to phosphorylate two serine residues at this protein, the inhibitor of kappa B. So now this inhibitor of kappa B, as it has phosphoserine residue, so to this phosphoserine residue will bind a E3 ubiquitin ligase, which is going to do the ubiquitination of this kappa B, and that is why this kappa B, the inhibitor of kappa B, it is going to expose to the proteasomal degradation. So it will undergo proteasomal degradation. So now this kappa B will be degraded due to the ubiquitination. So ubiquitination will be seen, and this will be degraded, releasing the NF kappa B in the cytoplasm. So now this NF kappa B, which is free, which is not bound to the inhibitor, to its inhibitor, this will expose its nuclear localization signal, and that is why this will migrate to the nucleus. So this will migrate to the nucleus, and it will act as an activator for the NF-kappa B responsive genes. So this happens when the different types of stress conditions are there and cell undergoes that, so that the NF-kappa B is activated. So this is the NF-kappa B signaling pathway. So the different types of genes that will be activated by NF-kappa B are, they can either encode, so the genes that are going to get transcribed, so the genes that are going to get activated by NF-kappa B, I'm listing them down here. So those genes can encode cytokines or chemokines, so that the neighboring cells, they are alerted of the infection. So it can release cytokines and chemokines, so that the other immune cells and fibroblasts, they can also accumulate at the site of infection to do their job and to neutralize the situation. So that is why release of cytokines and chemokines is important. Then it also encodes the receptor proteins. So activation of NF-kappa B is going to encode receptor proteins that are going to enable the neutrophils so that those receptor proteins that will enable neutrophils, so the neutrophils will migrate from the blood to the underlying tissue so that they can do their job, they can do the phagocytosis, and they can handle the infection. Also, the different types of genes that can be encoded by the activation of NF-kB, they include the INOS expression. So this INOS, okay, this INOS is the inducible isoform of an enzyme which is going to encode for nitric oxide. So the nitric oxide will be produced, and nitric oxide is actually toxic to the bacterial cells. So this is how the bacterial infection will also be handled by the NF-kB activation. But alongside all those genes, NF-kB also activates a gene which is going to encode for I-kB alpha protein, so that this is going to generate a negative feedback loop. So the once the NF-kB signaling gets activated, NF-kB will encode, it will activate those genes downstream that are going to tackle the infection, and they are also going to activate inhibitor of kappa B, so that the whole signaling pathway will be stopped. So for stopping the signaling pathway, a negative feedback loop is followed, and that is why NF-kappa B will be generating its own inhibitor once it is it is being activated in the ample amount. So this is how the NF-kappa B signaling is very crucial in these cells, and it is going to it will be important for the cell to be able to handle through the stress condition. So with this, we are done with the cellular signaling series. Okay, so I have a schematic for this whole signaling. Okay, yeah. It is here, so it is taken from Lodish, and it summarizes the whole lecture, the whole it's about things that I told you. So when there is bacterial or fungal infection, toll-like receptors are going to get activated, which are going to downstream activate this 3 protein complex. Or the TNF alpha can also activate this pathway. IL-1 receptor can also activate this pathway, or the other stress conditions such as ionizing radiation or viral infection. All of them, they are going to activate this 3 protein complex, and that is why the inhibitor kappa B kinase beta, this beta subunit is the converging point for all of those extracellular signals. So this kinase, this beta domain of this kinase is going to get activated, and this beta domain will phosphorylate the inhibitor kappa B. So this inhibitor protein will be phosphorylated at 2 serine residues. So here you can see this is the NF-kappa B, which is being sequestered by its inhibitor. So this NF-kappa B is inactivated, so it is in its inactive form. So this activated beta subunit will now phosphorylate the inhibitor protein of the kappa B. So it will phosphorylate, and that is why the 2 phosphoserine residues that are created, they will be able to bind to the E3 ligase, which is going to do the ubiquitination of this inhibitor protein. So here you can see the polyubiquitin chain is added to this inhibitor protein, which is going to now undergo proteasomal degradation. So this inhibitor protein will undergo proteasomal degradation, exposing the nuclear localization signals present at the N-terminal domain of NF-kappa B. Here you can see the NF-kappa B, which is a heterodimer having P65 and P50 monomer. So the nuclear localization signals will be exposed, which will result into the translocation of this NF-kappa B in the nucleus. So now when it's in nucleus, it's in the activated form, means it is going to work as an activator for the transcription of certain genes, which are going to produce enzymes, chemokines, inflammatory cytokines, adhesion proteins, and alongside all those proteins which are helpful for the cell to be able to survive the stress condition. This is also going to encode for the inhibitor kappa B alpha, so that the negative feedback loop is generated, means whenever the signaling gets activated, NF kappa B gets activated, it is going to encode its own inhibitor protein, so that it can go and bind to this NF kappa B again, and the signaling will be stopped. So this is how the this NF-kappa B signaling is very crucial in the cell. So we are done with the cellular signaling series. So I have covered almost all different kinds of signaling pathways that are present in the biological systems. If I missed any of them, you can tell me, you can suggest them in the comment section below. So I'll catch you in the next series. Till then, see you. Bye bye.